The first thing I really want to talk about, and is a topic that is very close to my heart, to our heart at Ercilia, is this concept of open science. Before talking about open science, perhaps I should take like 5 minutes to explain a little bit more about our initiative, Ercilia. As you know, Ercilia is a very small, not for profit, is a charity. Our mission is written here to strengthen research capacity against infectious and neglected diseases, especially in low and middle income countries. Our vision is a world with more egalitarian access to healthcare and research in biomedicine. So to achieve these quite big goals, as a small initiative, we focus on 3 pillars. The first pillar is this concept of free and open source. What do I mean by that? As you know, as you have learned, we do a lot of computational stuff. So computational assets can be private or public goods. We try to make everything public, so we share in real time everything we do. All the work that I have been doing this week or last week is already available for everyone to check in the internet. Why do we do that? Because we believe that, and it actually happens a lot, all of the time, we believe that science should be collaborative, and if I do some work, maybe someone else somewhere else in the world might benefit from it. Yeah, so we share code all the time, and we work with this spirit. We don't have licenses, like restricted licenses. Actually, everything we do is maximally permissive. This means that whatever we do at Ercilia, we share it publicly, and anyone can use it for free. Okay? No patents, and we put a lot of effort on reproducibility. And so we really believe that science should be reproducible, and we believe that putting code out there is the best way of actually double checking it. Yeah, because people can try it and people can come back with the issues that they find. So that's the first pillar towards bringing data science as a public good. Second pillar is in-country research. I mean, I don't think I have to explain this to this audience, but there is this tendency in the world of global health and in research that has been termed as helicopter research, where basically researchers from the global north might establish a collaboration with a researcher from the global south. They travel to the global south, they collect data, they take the data back to the global north, they analyze it there, and the scientific credit basically stays in the global north. This is something that should obviously be reverted, and we really work in this direction. So one of the things that we believe, and we actually do, is to do as much as we can research in country. So we actually, even if we are from the global north, we actually try to spend as much time as we can in the global south and actually support scientific projects that are already ongoing in the global south. The third pillar, also very important, is to establish sustainable collaborations. By sustainable collaborations, I mean, it's not just about doing a scientific project and then forgetting about it. It's actually about, as we all know, building capacity. So for building capacity, we need to do workshops, we need to do projects together, and we need to establish tools that are sustainable and well maintained. So this is what we try to do as much as we can as an organization. Okay. All right, so we are obviously not alone. And I realized that I didn't have time the other day to present this slide, but this is like a thank you slide to all the people that has been helping us over the last 2 years already. It's a lot of people, as you can see, so it's really collaborative work. Even if we look small, we have the chance, and we're very lucky to work with a lot of people, including H3. So of course, we would love to work with you also in the future from now on. So if you have questions, if you have ideas, if you just would like to learn more about us and see how we can collaborate, just reach out to us, either to myself, to Gemma, or to us as an organization. Okay. So the most important slide for me is what will happen after the workshop. And I have a few requests. Okay, so I would really like to, we didn't have time because it's been like very packed in terms of schedule, but I think it would be very interesting if you guys take the time after the workshop to actually reach out to us and tell us about your scientific research and whether we could be helpful in some capacity to provide some data science expertise in your research. If you are here, it's because you, I believe, have an interest in data science and machine learning, and therefore we hopefully are experts. So I think we can help. So please just get in touch with us, and we will, if you think it would be useful to facilitate a meeting with your supervisor or whatever that is, we can do that. We are happy to do that. It's part of our mission. Second point, actually, is not just about, I mean, you've been learning a little bit about machine learning, but it can be quite overwhelming. Actually, also as part of our mission, we do modeling on demand. So if you, and we do this as a philanthropic mission, right? So if you have a data set and we believe it's interesting and you believe it's interesting, and we believe there's chances for making a good model out of it, just tell us and we'll try to allocate some time to help you out here. You've learned a little bit about the Ercilia Model Hub. Gemma has been showing the Ercilia Model Hub quite intensively. I hope you got an idea of the possibilities that it has. I would recommend just don't do it alone. If you want to use the Ercilia Model Hub, if you think it can be interesting, just tell us. We know each other now and we can help you out. We can offer support. We can put models on demand, things that you might need. We can just include it there. We are all learning here. So please just let us know and we will work collaboratively towards this direction. Don't just try to use it and get frustrated, okay? And finally, very importantly, I realized yesterday that many of you didn't know each other. So you are building here a small network. So please get in touch with your colleagues in whatever capacity. Okay, I think that this is very important. It's so nice that we have people from different countries here, from different institutions, but with shared interests. So please try to use this opportunity to start building a network around it. Okay? All right. So let's talk a little bit about open science. Who has heard this word before? Okay. What do you understand for open science?
I think it's license that is not operators, creative commons, or their hand member, originally mainly applications that are for free. I'm not treating it because permission reference to the original.
That's one aspect of open science, making research and publications accessible to all, meaning no publication fees and no fees for accessing publication. Right? That would be one of the goals of open science. So ease of publication and ease of reading the publications. There's much more to it. Anything else that might come to mind about what an open science might be beyond open access, which is what we just mentioned? All right. So actually, it's quite a broad definition that it encompasses like many aspects of science, and I'm just listing them here. The first one is exactly the first one that has been mentioned now, open access. So if we are doing open science, this means that science is for everyone. Therefore, no publication fees. We were talking about it yesterday at dinner time, and no paywalls for accessing publications. Same happens with data. If we are doing open science, then data should be available to all. Same happens with code. If we are doing open science and if we are doing machine learning, code, software should be open source, available to all. Same happens with hardware ideally, right? If we are doing open science and we want everyone to be able to do, let's say, machine learning, infrastructure and hardware should not be a limitation. So ideally, in an ideal world, we should all have access to hardware infrastructure. We've been having some access to hardware thanks to Google Colab, for instance. You didn't realize maybe, but the code that we were running was not running in your computer, was running in Google's computer. So that's an example of open hardware. Yeah. Open infrastructure. pretty much the same idea. Open methodology. So if we are doing open science, it's very important that we report protocols so that people can reproduce them. And that's an essential pillar of open science. Open education, right? Education should not be a privilege. If we are doing open science, everything we are talking about should be accessible to all, right? There's some instances of very interesting and good courses that are open in the internet, as you may know. Open peer review. There's a whole thing, as you know, peer reviewing. Ideally, in an open science world, the reviewing process should be open. This means that everybody can see what's going on during the reviewing process, and everybody can see in full transparency why a paper was accepted or rejected. And open social engagement. This means that building networks without hiding this fact. We all know who are our partners, we all know who are our collaborators, and we share this information to the world. And so I think that those are the basic principles of open science. We are clearly far from that, but I think it's a nice goal to have. So can we bring open science to drug discovery, where, as we know, traditionally drug discovery has been locked behind patents and behind intellectual property constraints, understandably. So yes, there are some initiatives that I'm listing here that are doing to some degree open science. We all know Medicines for Malaria, great initiative, right? These people are designing compounds, they are making results public, and not even that, they are even distributing compounds in these malaria boxes or pathogen boxes that they are. So that's a great example of open science. There are others, even from pharmaceutical companies, for instance, the Tres Cantos Open Lab Foundation by GSK in Madrid. That's also a good example of open or almost open science. In the field of drug discovery, even I think it's in Australia, there is this COAT, which is for escape pathogens. Quite a great initiative. You can send compounds there, they will screen them for you, et cetera, et cetera, et cetera. Of course, the COVID pandemic has basically highlighted the need for open science in this world, and therefore there are many open science and collaborative efforts that have emerged thanks to COVID. So can anyone say or answer, why do we think open science can have some advantage in drug discovery? Any ideas? Any thoughts?
One thing that comes to my mind is it helps to avoid several people repeating things that someone else is doing elsewhere and evolving in the same stage. But if you know someone is doing this and they've advanced to this level, you can now think of, I think, the next stage so that it's advancing and to reduce a lot of repeating the same things that when someone discovered doesn't work or has this issue.
Avoid overlap, right?
Yes.
I think it saves the time. The drug discovery takes a long time. Companies try to do it with potentiality and protecting everything. They actually take time. An example of open science in drug discovery, they said, was just a lot of open actually to short time to discover drugs and decisions.
Okay, there's clearly disadvantages to it, right? So obviously, like pharmaceutical companies and any company really needs to make profit, and therefore we should understand this fact. And I think that as researchers in open science, we need to find the best ways of actually making this also attractive to pharmaceutical companies and people who actually want to get profit, which is perfectly understandable and there in the fact that we will see some instances later of how we are trying to do that. Any idea of how artificial intelligence might support open science? I'm running out of battery. Yeah. Any idea of how artificial intelligence can support and benefit from open science?
Open science, like as we discuss about open science, it's open accessibility, open data. The moment you have a lot of data from it, everyone is accessible to the research articles and all these things.
Yeah.
And they are all putting their efforts to produce some qualitative data. So that data can be used by the artificial intelligence to filter out and to support the drug discovery to progress further.
That's a great answer. So more open science, more data for everyone, better models, always. And therefore, we are, of course, in favor of this field because I think it ultimately benefits the field of machine learning and data science in general. All right. So don't run away. This is how open source, open science works typically in organizations that are virtual like ours. Yeah, so we do code all the time. Some of you already knew how to code. Many of you were new to it. If we are doing open source and open science, one of the most important resources we need to consider is GitHub. Okay? GitHub is a global repository of code. So you go to GitHub and you will find a lot of code for a lot of problems, right? Many publications, as you will see, if you start reading machine learning publications, they have a GitHub link at the end, and you can access that, and then you can find the code corresponding to the publication there. So basically, each group or each lab or each company has a GitHub organization profile. Yeah. This is the GitHub organization profile over Cilia. And as you can see, we have almost 150 repositories. That means we've been doing quite a lot of work. 150 tools that we built in the last couple of years. We have 3 main projects, like really big projects that we are actually monitoring and where all these repositories fall. We have some people that are part of the organization and that help us in some capacity. And here you can see all the repositories. We are highlighting some of them. The most important, the one that you guys have been using, is this Cilia repository, which is to access the Cilia model. So you can visit an organization profile, and just by looking at how much work they are doing, how much activity do they have in each of the repositories, you can kind of see how active and how healthy, in a way, this organization is. All of this is public, of course. All of the repositories here are or will be public. So let me go back and highlight here one of the latest repositories. We just published it, I think, 2 weeks ago, called ChemSure. And the reason why I'm going to highlight this repository now is because ChemSure is hopefully solving one of the main problems we find when trying to bring pharmaceutical companies into the open science landscape. Okay, what's the main problem that you think, or the main concern that pharmaceutical companies might have in becoming completely open science? Ideas? IT, right? In particular, the main asset of a pharmaceutical company is clearly the chemical library that they have. If they reveal this, then there's no business, right? Yeah. So, I mean, it's not realistic to think that they will just publish that in the public domain for the greater good. It's not going to happen. So we need to find alternatives to it. So we've been thinking about this problem quite a lot, and the alternative we found, and we hope it's going to find some adoption. This is a project that was already funded by Merck, so there is some interest. The pharmaceutical company. The idea is, we've been talking about this cycle, right? The machine learning cycle. A machine learning tool, we've seen that it can be very useful, especially when we bundle it like that. So we just put an input, a drug, as we've been doing during the course, and we get an output, targets, admin properties. activity against a pathogen, whatever that is. Inside this machine learning model, there's a lot of steps that have been done. And the main and critical one is the central one, which is the raw data. We don't want this to be revealed, because if this is revealed, then basically the pharmaceutical companies would compromise their RP dramatically, right? So what we are offering actually is an encryption protocol. So what we think is that perhaps it would be enough if we just ask the pharmaceutical companies or offer the pharmaceutical companies to train a machine learning model based on their data on their premises, so in-house. And then they just distribute an encrypted version of the model, right, to the public domain, so that us researchers can use this model for our research. Remember, this model has learned the patterns from their data, so it's a very informative model, without ever seeing what was the data that was used at training them. Yeah. We believe that this is an interesting approach towards unlocking, to some degree, the data that is currently sitting in archive in pharmaceutical companies. Yeah. And Chemshore, this library, Python library that I was highlighting, is doing exactly this step, encrypting everything so that no one ever will be able to actually reverse engineer the molecules that were used for training this model. Yeah, this sounds very easy, but it's very complicated technically. I think we are there already, and it's just a matter of finding good adopters in the pharmaceutical industry. Okay, good. We are not the only ones doing this or similar things. Actually, this is something that is happening a lot in the field of medical data, and not so much perhaps drug discovery data, but certainly in collaborations within hospitals. Of course, hospitals don't want and should not reveal the identity of their patients, and therefore, when we are thinking about collaborative efforts between different hospitals, we need to have some encryption protocols so that the partners in a collaboration do not see each other. Okay. So there are, this is a Nature paper, very nice, from last year. There have been, people have been suggesting different approaches to do exactly this, to train models collaboratively that can learn from the data from each of the institutions without ever revealing to anyone else outside the institution itself the identity of the, in this case, the patients. Yeah. There's different approaches for achieving that. I'm not going to go into the details. One of them is called, has a very nice name, called swarm learning. And in swarm learning, basically, you have like people are distributing the tasks between different institutions, and it's like a collaborative training effort. I'm not going to go into the details today, but I really recommend like reading this publication because it's good food for thought. Okay. All right. So let's switch gears now and talk about, people have been asking a lot about generative. Okay. So I like to show this picture from 2018. This is a piece of art, and that was sold in an auction in New York, 2018, for $800,000. This, okay. So why do you think this has such a great value? Does it have aesthetic value? Really not. Does it show like future skills from the painter? So why is it so interesting? Right. It was generated or invented by computer. Yeah. So there's no artist behind that. The author of this is an algorithm. Yeah. So this is the first instance, or one of the first instances of art generated completely de novo without the intervention of a human artist by a computer. Yeah. So that's a landmark achievement. Since 2018 on, the field has gone crazy. Like it's becoming every year, we have a breakthrough in the field of generation of images. Okay. So today in 2022, there is this algorithm called DALI, who you might have seen it in the news. Yeah? Developed by a company called OpenAI, which is now owned by Tesla, basically. That basically the way it works is you write the text of what you want, and it's going to generate an image for you. Yeah. So let's try. For instance, an astronaut riding a horse. You write that to the tool. It's an AI, okay, like a tool like in the Ercilia Model Hub, actually, and you get it. Okay. So, I mean, it's remarkable. And first, the quality, and also like the capacity of merging objects that belong to different fields in principle, quite remarkable. Okay, we can try. I don't even know if my use of English here was good, but a ball, like things that don't make sense. A bowl of soup with a bull monster. Yeah, I don't know if this makes sense at all. Let's see what DALI says. Yeah, so it's really remarkable. Yeah, this is done. This takes literally 5 seconds to compute in your browser. Okay. I'm going to try one that I really care about. A nomad city full of threads that go from door to door. This is a metaphor that gives name to Ercilia, which is our initiative. This is a description of an invisible city in a book that is called Invisible Cities by Italo Calvino. And basically, Ercilia is a city where people throw threads from door to door to denote relationships of friendship or father, son, things like that. No, so and the nice thing about this city is that when the city becomes like too populated because there's too many threats, they just abandon the city, they go somewhere else and start over again. Yeah, so I like the metaphor. Let's see if DALI is able to suggest things. This one, yeah, don't like it that much. This one is perhaps a bit more interesting. If I ask to draw like a kid and do things like that, or like an impressionist painting, or more psychedelic. Yeah. So it's remarkable what these kind of things can do. For instance, I was trying to come up with a cover for the algorithm for privacy preserving, yeah, AI that I've just shown, Chemsure. So I asked DALI to build a cover that is called Whispering Molecular Secrets, yeah, and do it in modern art. So it suggests something like that. Yeah. So it's cool. Now I have a cover for my album. Yeah. So of course, this is working in the field of image generation, which clearly has a potentially a lot of economic return. We are not artists, we are chemists. So what we really want is something like that. Yeah. Yeah. So I wake up in the morning and this is all I think about. I want a novel, cheap, safe, and potent antimalarial compound. I want this algorithm that is going to give me the answer, right? This is what we want. Of course, we are not there. We are not there for many reasons. Of course, we are not there, and it's going to take time. But I'm convinced we will get there. So I was checking the other day, there is this paper. It's just a preprint by Microsoft. It's called Translation Between Molecules and Natural Language. Exactly what we want to do. Natural language, what we write, molecules, what we understand as chemists. So for instance, the first example of the paper is this one. The molecule I want is an 18-membered homothetic cyclic peptide, which is isolated from this species, and exhibits antimalarial activity against this strain of the parasite. It has a role as a metabolite and also as an antimalarial, and like a rough description of the molecule. Yeah. This algorithm, published as a preprint, is already able to understand this text and say, oh, hey, maybe this is the molecule. So we are not there yet. It's not the perfect result, but we are getting there. Okay. So it's possible that in the near future, as chemists, we will have to interact, or medicinal chemists will have to interact with this kind of tools in our favor, not to lose our jobs, on the contrary, in our favor, to get better ideas, yeah, of the compounds that we might want to synthesize. So this is where we are, I think. We want to generate molecules based on a target activity. And this is what we want. We want to invent molecules that will do something that we want. Yeah. So we've been talking a lot about, remember, that was supervised machine learning. We know activities. We train a model, and when we come with a new molecule, we will predict the activity. That's supervised machine learning. When it comes to generative models, the other way around. We have an activity in mind. We want to predict the molecule that is going to have this activity. It's clear that the problem is much, much more difficult. It's not so easy. This is why we are not going to do it quite a lot hands-on today. We'll do a little bit, but not a lot. Okay? It's a difficult problem, but it's within reach for some cases already. Yeah? And it's going to get easier and easier and easier because there's literally an avalanche of papers trying to do that exactly this today. Okay. So let's get into it. Usually, we can think about this problem of generating small molecules in two modes. Yeah, and this is like jargon of reinforcement learning, which is the third column, if you remember, of the types of algorithms that we have. Those are the generative models typically. We have exploitation mode and exploration mode. Okay. So let's just start with exploitation mode. Let me put it in your words. This is hit to lead optimization. Yeah? So if we think about a bioactivity landscape, in a hit to lead optimization, have a hit that is quite good, but it's not optimal. Okay. And what we want is to exploit this hit. So we want to invent molecules around this hit that are not too different from my hit, of course, but have better properties. Yeah. That's the exploitation mode of the method. The exploration mode, I believe it's would be library design, right? You want to design a set of more explorative compounds so that you can come up with a higher diversity. Yeah. So typically in exploration mode, you might or might not have an initial idea, but usually this initial idea that you have, it's far from a hit, just something there. It's not really active. And what you want to actually have is more molecules to screen that are as close as possible to your activity of interest, but perhaps not so optimal. You just want diversity. Yeah. So these methods, we can make them work in two modes: very focused or optimizing for diversity. We'll see some examples now. So for instance, let me prove that this is not something that is just science fiction. You can see here a hit for antimalarial activity, basically given to us by this open source malaria consortium that I have just mentioned before. And the task was to generate more hits, more candidates that look like that, more or less, with the method. So at the time, we only had this computer, so we did it with this computer. So it's feasible. And you can see here how my generative model is generating more and more and more and more candidates around this hit. The more blue this becomes, it means the more active I predict the molecule to be. So it's like I'm going up in the predicted activity hill, no? As you can see, there's many, actually as many as I want. I just have to wait, okay? This was done with a reinforcement learning algorithm called reinvent 2. So if you are ever interested in doing some generative models, I would recommend that you check this reinvent 2 or 3 now. I have the 3 now article. It's quite good and powerful. Yeah, as you can see here, there's a moiety that is conserved. And then the rest, I'm basically just optimizing the branches of this initial model. Great. So after this round of generation of molecules, I think we generated like 100,000, something like that. I'm just putting here some snapshots. The more blue, as I said, the better I predict them to be, but I believe that all of them might be interesting to some people. Yeah? So that's a lot of information for a chemist to digest. So accompanying this generation of molecules, we always give like some sort of visual tool so that chemists can explore it. These are the 100,000 hits that we generated over an afternoon with this computer. And here, basically, I'm highlighting in red the regions that we believe are more interesting, meaning more active. So if you are a chemist, I can give you this map. Each dot here in a map is obviously a molecule. Dots are nearby if they are similar molecules, so you can navigate this map easily. And of course, what you want to do is to focus in these red regions, and let's see if you find something interesting. So you can obviously do it, and as I said, each dot is a candidate. You can recognize here the constant scaffold and then the 2 branches. Yeah. So a lot of chemical matter that we can generate with computers relatively easily. That's just an academic exercise. So what we did afterwards was to actually go back to open source malaria and say, hey, we have 100,000 compounds. We obviously need to filter them. Let's do it together. And I will show you now how we did it. But at the end of the day, at the end of the pipeline, we selected, I think it's 25 compounds that they have synthesized, and they will test them. I think they are actually testing them this month, at least some of them. As you can see, even if they were generated completely by a computer, this is not crazy chemistry. This is reasonable. Yeah. So remember, open source. We need to make this available to everyone as soon as possible. So you can see here how we basically document all of this procedure publicly. So in GitHub, open source malaria has obviously a GitHub organization profile, and there's always issues. And in issues, there is discussion. It's like a forum, like a chat. So I opened an issue 1 year ago, a long time ago, and I said, hey, Matthew, the PI of open source malaria, I think we've generated some compounds. You can find them here. Please have a look. And then we start a conversation. I actually show them some analysis that we did so that to help them understand the results. Exploitation and exploration, meaning exploitation is this region here, yeah, of the initial compound. Exploration is the red region. Yeah. So I was like, hey, we have exploration hits, and exploitation hits. Please tell us what's more interesting to you. More boring conversation, blah, blah. They found some interesting candidates, but they had some problems in the synthesis. Someone else jumped in, etc., etc., etc. So all of the conversations that we are actually having on a daily basis, for us, they happen in the public domain. Okay? This is the spirit of open source. So more and more conversation. And at the end of the day, we came up with this filtering procedure. It's just a filtering procedure. We could have decided another one. We generated, that was lying, not 100,000, almost 500,000 compounds in one afternoon. And of course, we had to filter it down to the 25 that I was showing. So we were like putting different filters that all make sense. For instance, this triazole pyrazine core seemed very interesting and quite conserved, so we wanted to maintain it, and this removed already some hits. We defined some desirable physicochemical properties like log P, etc. And this helped us also filter it. We optimized for synthetic and retrosynthetic accessibility of the compounds, which can also be calculated, as you know, with machine learning. We made a predictive model, and we took of malaria, and we took the ones that were better hits. We eliminated redundant molecules, etc., etc., etc. We used this MIPE model that Gemma was showing the other day to further filter, etc. Yeah, so it was like a filtering procedure that we decided at the end of the day. From an initial collection, we came up with a realistic set. So this is how it works usually. You expand, you generate a lot, and then you filter. This is how it goes. Okay? People often get frustrated with generative models because they expect that all the molecules that are generated will be valid. That's not the case. You really need to do this filtering afterwards. Okay? Great. So don't run away. There's different families of generative models, and it's really out of the scope of this workshop to understand, but I just want to give you the words, okay? First family, you will see it a lot, is called variational autoencoders. Okay, intuitively speaking, what's happening in a variational autoencoder is that you have an input molecule, let's say a hit. You compress it with the descriptors that I was showing. Once you have it compressed, you add some noise. You add some noise, like, you know, some noise, and then you decompress it. By adding this noise, your molecule is changed. And when you decode it or compress it, you obtain a new molecule. Yeah? If you add just a little bit of noise, the molecule that you will add is going to be very similar, right? If you add more noise, the molecule is going to be more that different to your input. Intuitively speaking, this is what's happening here. That's the easiest one to understand. A lot of variational autoencoders are out there, and it's quite interesting. They are quite slow though. This is also fun. Generative adversarial network. In this case, it's actually two machine learning models, and they are, oops, sorry. That's the word of time. They are competing with each other. So there's a model that is generating molecules and is trying to fool the other model. The other model needs to say whether this molecule looks good or not. Yeah, it looks real or not. So one model is trying to fool the other, and the other is trying to learn to discriminate between the two. By doing this procedure, you come up with a very efficient way of generating good molecules. Okay, because the model that is discriminating is challenged by the other one. So it needs to actually become very good at discriminating between good and bad molecules. Okay, that's intuitively what's happening. Reinforcement learning, the most widely used now, is this idea where you have a generative model, and then you have agents that evaluate your candidates. And these agents can evaluate the activity, the accessibility of the compounds, they can evaluate the price, the log P, anything you want. Yeah, actually more than one thing at the same time. And therefore, this agent will say, hey, you did a good job or not? You did a good job or not? And the model needs to learn to do a good job most of the time. Yeah? And this is called reinforcement. By doing so, you come up very efficiently with good sets of candidates. And then there's recurrent neural networks. I would say that they are already too old now, and hybrid approaches. We don't really care. So it's variational autoencoders, generative adversarial networks, and reinforcement learning. That's the keywords. So if you find a paper that mentions this, don't run away. Intuitively speaking, it's quite you should find, okay? Because it's not such a difficult thing to understand, at least at a high level. Of course, the open source malaria thing has been quite successful. For us, we've been trying this in other contexts, for instance, in the open source antibiotics challenge. In this case, it's not for malaria, it's for Staphylococcus aureus. And in this case, what's interesting is it's not a wholesale assay of antimalarial activity in that case. It's actually, they really have a target of interest for which we have struck. So in this case, the generative model procedure is a bit different because we actually know the binding site and we need to generate molecules that will accommodate in that binding site. Yeah. So the way we score our candidates is by seeing how well they dock in this binding site. Yeah, we did that. Again, generated, I think in this case, it was only 50,000 compounds. And we selected at the end of the day 150 that are purchasable, and I think they will test 10 of them. Okay, here you can see a snapshot of some of the compounds that we generated. In this case, we run this in exploration mode, not in exploitation mode. This is one of the example compound candidates that we have that docks quite nicely in the binding site of this enzyme of the Staphylococcus aureus. Great. So what other things you can do with these generative models? As a lot of researchers have said, this is one of my favorite papers of last year. Synthesis on a chip, they call it, on-chip synthesis. In this case, you have a generative model with a limited set of reactions. It only allows and accepts molecules that you can synthesize with a limited set of reactions. Reactions that can be done with the microfluidics platform, right? So what do you achieve by that? Well, you have an automated system where your model generates candidates that can, by definition, be synthesized with the microfluidics platform. Therefore, you have a virtual input, you get a real output. You can synthesize directly your candidates. This is great. And they actually have shown in this paper that they can synthesize some chemical series quite efficiently with this kind of technology. The limitation, of course, is the limitation of the microfluidics platform itself, which is limited to a certain number of synthesis steps and reaction types. But conceptually, it's a very interesting technique. Actually, since 2018, published in Nature, there has been also a revolution in retrosynthesis planning assisted by AI. And I don't have to explain anyone what retrosynthesis is planning. The idea is that you have a compound that you want to synthesize and you want to predict which are going to be the best synthesis steps. So in this case, these models are trained on reactions from the literature, and they try to come up with a synthesis route that is short enough and feasible enough, starting from a set of building blocks. This method was kind of good, but the field has become more and more proficient today. Yeah, so an interesting thing to do, and we actually do that, is to couple generative models with retrosynthesis planning. So you only accept things that your algorithm predicts that are going to be easy to synthesize. Okay. Great. So all of this generation of molecules has created in result huge chemical space, at least virtual chemical space that is accessible today. So that's a nice summary of the libraries that we can access today as researchers. Is quite crazy if you think about it, if you look at the numbers. So PubChem, which is the one that you probably know, 10 to the 8 compounds, that's already quite a lot. Yeah, it's around 100 million compounds or so. There's these virtual libraries that have been generated with the simple generative models containing molecules with only 17 atoms, I believe. 10 to the 11. I cannot understand this number anymore. Yeah, approved drugs is only 10,000 or so. This is accessible publicly. Commercially, I'm sure you know all of these companies, Sigma for sure. molecules that we can buy. Enamine, for instance, there's this real database, they call it real database in Enamine, you might have heard of it. Those are libraries that are called synthesis on demand. They don't have the molecule, but they know they can build it in less than 3 weeks. So they use the generative model to actually design those libraries that have billions of compounds. Yeah. What's great about this is that if you find a compound there, you can synthesize it. Actually, you can just order it and you will get it in 3 weeks. Okay. So those are called ultra large libraries. And this is what's accessible to us as researchers. But if you think about pharmaceutical companies, I mean, 10 to the 20 merge. Yeah, so it's a lot, which even begs the question, why do we need to invent more molecules? I mean, there's so many. There's really so many. So I do think that even if generative models are very interesting, and they are, it's also very interesting to consider whether we can identify in these purchasable libraries, ultra-large libraries, whether we can identify interesting chemical matter there. Yeah. Because those are molecules that nobody has synthesized yet, but they are easy to synthesize and purchase. So it makes it very convenient. And actually, we are doing this all the time in our research. Yeah, we look for molecules that we can buy easily. Yeah, so we don't have to invest a lot in the synthesis, at least in an initial screening step. So let's talk about a little bit about this Enamine library. So how they build these libraries is like it used to be called combinatorial chemistry before, if you remember, no? The idea is that you have building blocks and you have a limited set of reactions that you know you can do in your company. So basically you can combine these building blocks and say to your clients, these is all the molecules that I can offer. Yeah, because I have the building blocks, I know how to do the reactions, therefore I can offer potentially all of this chemical matter. As I said, Enamine today, I think they have like about 20 billion compounds that you can potentially buy. That's a lot. That's a lot of molecules. Can we treat this with computers? So yes, we can. And that's one of the great things about machine learning, that it's really fast compared to traditional chemoinformatics tools, let's say. So for instance, there's been a stream of papers actually tackling this problem of ultra-large virtual screening in different tasks. This is a nice one published in Nature this year. In this case, they took the Enamine library. This is why I was highlighting it. At the time, the Enamine library had 11 billion compounds. And the question was, can I dock 11 billion compounds against my target of interest? If you have to do conventional docking, this will take many years, right? Many years. So you cannot possibly do that. But you can accelerate the procedure a lot with AI. And conceptually speaking, and I'm not going to go into the details, what you do is you just dock a small fraction of the molecules, and based on those results, you train a machine learning model that is going to be able to predict for the rest. And then you do this iteratively. So actually, you are not doing docking for all of the 11 million compounds, you just do docking for a subset of them, and the rest, you predict them with machine learning. This is intuitively what these nice papers are doing today. And I think that this is a really smart approach and really brings together the field of structure-based drug discovery with the field of machine learning. Yeah. So it's really something to consider, to actually not screen just 100 molecules or 10 molecules or even 1,000 molecules if you're lucky, but actually screen billions of compounds that are already purchasable. You might find interesting kits there. All right, so my last slide, because I'm already over time. Recap, what we've learned. Supervised machine learning, that was the second day. When do we do supervised machine learning? When we have labeled data, and we have two types. You all know this already, classification and regression. Here I'm showing a classification task. I want to find a model that is able to discriminate between actives and inactives. Yeah. What applications do we have in unsupervised machine learning? Bioactivity prediction, what Gemma showed you. AIML properties, as we did yesterday with Matthew and Lisa. Yeah, great. Unsupervised machine learning, that was the Jason's tutorial in the first day. In this case, we don't necessarily have activity data, but we can do many things anyway. We can cluster compounds, we can do similarity searches, and we can do 2D projections. In this case, I'm doing the 2D projections and then clustering. So I'm discovering different families of compounds. Why do I want to do that? Well, to visualize chemical libraries, discover series, discover groups of non-redundant compounds, etc. That's very, very useful in understanding your chemical space. Today, we've seen a little bit about generative models and reinforcement learning, which was the missing pillar. Why do we want to do that? Well, in exploration mode, we want to do it to design libraries, compounds for screening. In exploitation mode, we want to do it for hit-to-lead optimization. Okay?